So welcome to the course of Fundamentals of General Immunology, and this course is basically designed in order to give a proper understanding about the immune system, how the immune system behaves, how the body response is towards a pathogen would be, and what are the bottlenecks which are required in order to understand the immunity when it comes to the protection and overall well-being of a human being. So this overall course structure is divided in such a way that we'll have a lot of modules, and there are around 15 modules and about 35 lectures. So this is the first lecture in the module 1, and the module 1 entitled is Immune System as a Dynamic Defense Network, and the lecture 1 is Evolutionary Logic and Functional Architecture of the Immune System. So let's begin with lecture 1, which is Evolutionary Logic and Functional Architecture of the Immune System. So if we look into the threat and the human body is a very complex structure. It has multiple organs, multiple systems, and each system is controlled by a variety of regulatory mechanisms. These are starting from a basic structure which are from the gene level, and also from if we start from the central dogma, the DNA, which is converted into a specific kind of messenger RNA sequence which is called transcription, and then again this messenger RNA can be encoded further into protein, that's called translation. So this complete central dogma makes the whole system very complex, and the human body itself is made up of multiple forms of these regulatory structures. So let's understand the whole system. So if you see in this slide here, so if you see, this is the complete human body, and if I go and read the content, the immune system is a dynamic surveillance and defense network built to detect and eliminate infectious agents while preserving the host tissue. That stands for the immune system can always do a dynamic surveillance towards the entry of any pathogen or entry. When I say pathogen, the pathogen stands for a virus or a bacteria or a fungi or a parasite. So these are the microbes, and if you grossly look into the term, people also say microbes, and then it is a macrobes. Although these are not the term which are properly used in immunology, we will try to stick our discussion to microbes. And the microbes are something which are visible only under the microscope, very tiny microorganisms which can evade the host immune system to produce a disease condition in human body. So ideally, to build up and detect the immune system, we have a very cascade, very synchronized system of the immune regulatory. So there is a constant threat from the environment side. So the human body is exposed to the environment every day, and these could be a heat stress, it could be a environmental moisture effect, it could be anything which are there in the environment, and the major entity which decides the whole immune system more complex is that pathogens, and these pathogens are what we just now discussed. These are all 4 microbes: virus, bacteria, fungi, and parasites. So we do have a counter pathogens that continuously evolve to host the immune system. That means if we have our immune system robust against a pathogen also try to learn lot of techniques in order to evade the host immune system, and some of the mechanism which we'll go in detail, discuss in detail later on. Some of the mechanism could be the change in the gene structure. So the pathogen might change in the structure of a gene so that the protein will be changed, and therefore the body thinks that these pathogens has been encountered to the body system earlier, and therefore the immunity can be produced. But then because there is a change in the protein, body feels that these pathogens are kind of new, although these are the same pathogen which are mutated in the form of a different organism because of the change in the gene structure. Therefore there is a rapid adaptation of the pathogen, and of course there is a internal harmony. So what is generally done is to maintain the internal harmony, the harmful microbes must be distinguished from the harmless ones. So giving an example, in the gut system of a human body, we have lot of small tiny bacterial population, and these bacteria are very useful for the maintaining gut homeostasis. So gut microbiota is very important because these pathogens, not pathogen, these are the bacteria which are present in the gut, can synthesize lot of essential enzymes and some regulatory metabolites that's very useful for proper maintaining of the human body. So body should not respond an immune response against those small tiny microbes which are present in gut. So we call it gut microbiota, and if there is a disturbances in these gut microbiota, then essentially one can realize that there is a problem in the gut homeostasis. So people may suffer from a disease condition which are actually alarming, and that's simply because the useful bacteria which are there in the gut is not present. And one can always visualize that when a person is undergoing an antibiotic treatment against a bacterial pathogen, then what is happening, that antimicrobial or antibiotics such as ampicillin doesn't differentiate between a useful and the harmful bacteria, and therefore it can flush out all the bacteria species from the body, which includes the gut useful bacteria as well. And once there is a complete flush out of the bacterial system from the body, essentially you start getting certain kind of problems such as gastric acidity, also the bloat and other problems, and therefore you need to replenish the bacterial gut microbiota by using some probiotics. Probiotics are the solution which are there in the medicinally present in the market, which contains all the useful bacteria to repopulate microbiota. So the next one is the host pathogen arm race. So the immune system will not directly appear, right? It gradually appears, and it evolves gradually, and there is a whole bunch of evolutionary mechanism which has been documented that says that the pathogen is evolving, host immune system is also simultaneously evolving, and we are adapted into the system. So I'll just give an example, two examples, how this pathogen moves and how the body immune system also moves in this ladder. So first come to the pathogen. So the pathogen, as I just mentioned earlier, that there is a mutation. So the viral mutation enabling immune evasion. What it stands for, that when you have a mutation very categorically in a gene that leads to a deviation in the protein. So the body doesn't recognize this protein as what it recognized earlier, and therefore the pathogen may escape from the host immune system. So there is a constant threat that is happening because the pathogen wants to evolve themselves by doing certain mutation in the proteins, and these mutations are sometimes very deleterious. We have documented COVID-19 pandemics in 2021, and which started in 2019, which basically tells that although this virus is not a new virus, coronavirus is actually a very well established virus, but because of the subtle mutation in the genome, the virus came out to be very, very harmful to the human population, and our immune system doesn't recognize it as a one which already have the immunity against the earlier coronavirus in the human body, and therefore there was a pandemic, huge pandemic. And if we categorically see the COVID-19 virus, there is a mutation in the spike protein. So there is one protein which is immunogenic protein. When I say immunogenic protein, which stands for one of the pathogenic protein which body recognizes as it as a foreign and produce an immune response. So the spike protein of coronavirus is important protein, and therefore one has to think about making a diagnostics or vaccine using this spike protein. So the COVID-19 SARS coronavirus 2 modulated its spike protein in such a way that it can escape the host immune response. So this is one example in terms of virus. The second one is quoted here, where you have a bacterial secretion system. In the bacterial secretion system, there are many secretory system present in the bacterial system, and therefore bacteria learn how to make this secretion system little differently engineered in a way to evade the immune system. And therefore, for example, if we have an E. coli secretion system, this can do certain kind of a toxin production which is harmful to the human body, and therefore subtle deviation in the toxins could lead to large deviation in the pathogenicity of the bacteria. So these are the two examples of pathogen that is evolving gradually in order to sustain themselves in the human body and to escape the immune system. Now, what is a counter argument? What human body does? Human body does exactly the same kind of evolutionary biology in order to fight with that change in the genome, such as mutation and the secretion systems. So what it does is there is something called a pattern recognition receptor, which is here PRR. So this PRR is the very specific signatures which are present in the host cell that identifies a pathogen specific proteins. So we will learn it a little later that what do you mean by pathogen associated molecular pattern, which is called so this is pathogen associated molecular pattern, PAMP, and this PAMP is actually a part of pathogen, and this pathogen is specific or associated molecular pattern can bind to the host specific PRR. So pattern recognition receptors, and these pattern recognition receptors are present in host cell. So this is host specific and this is actually a pathogen specific. So this is pathogen specific. So these two are always in the constant fight, and if PRR and PAMP binds to each other, then they can easily neutralize each other. Therefore, what it stands that when a pathogen is mutating, the PRR, the host pattern recognition receptors, are also evolving in order to bind to that mutation change, and therefore there is a constant change in the human immune system. The second is the host counter, and this is the development of a specific, highly specific memory capable adaptive immunity. So we will learn gradually there are two different arms of the immunity. One is present right from the birth, which is called innate immunity, and the second is which we are gaining, which we are experiencing after exposed to the environmental antigens or molecules, and these are called adaptive immunity. And this is a very, very complex mechanism, and how this adaptive immunity can recognize the molecule as a foreign and store the memory, so that in future, if at all this kind of pathogens are coming into the body, they can recognize it because they have already prior experience to those pathogen. They can immediately jump into the memory and synthesize some of the molecules which can destroy the pathogen. So these are very synchronized way that pathogen is also evolving, and simultaneously the host immune system is also evolving. So let's try to understand very important part which we will discuss in this whole course, and that's called blood. So this blood is actually defined as fluid connective tissue. So this is actually a liquid, and since it connects to the two different cell type or the organ, we called it connective, and since it's a collection of cell, we call it tissue. So the simple definition of blood is a fluid connective tissue. So we generally deal with the cellular or the soluble component which are present in blood system. So the understanding blood is very important. So if you just look into the panel which is here, that the blood circulates into this small tube-like structure which we call blood vessels, and these blood vessels can carry multiple different kinds of cells such as RBC, red blood cell, WBC, white blood cell, and other small cells which are very important for the immune regulation. So if we look into the blood vessels, there are two kinds of blood vessels. One is called veins, and the other is artery. And if we looked closely, the veins are the one which carries the impure blood, and the artery are the one which carry the pure blood. Except there are exceptions, and all the biological system is full of exceptions. So there are pulmonary veins and pulmonary artery. So pulmonary veins are the one which carry the oxygenated blood, and the pulmonary artery are the one which carry the deoxygenated blood. So these are very common, say, concept which one has to know. Apart from blood vessels, we can also look, these are the small venules kind of structure, small, we call it capillary, and this capillary are very fine membranous bond system which can supply the blood to the cell level, very fine, small, specific tube-like structure which can communicate to the cell level. So these blood vessels actually carry all these specific cell type in a very constant fashion, and if there is a dysregulation such as high blood pressure or the low blood pressure, we see a deviation in the circulation of this blood flow. There are two other terms which we have to understand. One is called plasma and the other is called serum. So these two are very, very important and critical components of the immune system. The plasma is actually the liquid part of the blood when we settle the blood. What it stands for? The blood is actually a fluid connective tissue, as we discussed. When we add any anticoagulant, the blood won't coagulate. Otherwise, if we take out the blood in a small tube, it will immediately clot. If it will immediately clot, we won't see the fluid pattern of the blood. But when we add the anticoagulant such as EDTA or heparin, these are two specific anticoagulant. Heparin is the one which can use for making the blood in a fluid form, and EDTA is a chelator which can chelate the calcium ion, and therefore this won't allow the blood to coagulate. And once we have a blood added with an anticoagulant, that means blood won't coagulate. If we spin it down, the blood cell will settle down in the bottom, and what fluid part we will get is what we call plasma. So can we say that these are the liquid part with clotting factors, antibodies and signaling molecules. So these are all parts which will be present in this specific plasma content. The second is serum, and just opposite to the plasma. So plasma is without any coagulation, but when we take the blood without any anticoagulant, the blood will immediately clot. And once the blood will clot, the fluid portion, which will be a very small amount of yellow straw color fluid, and that will be present in the top of the clot, and these are called serum. And serum is full of antibodies which we will study very often in this course. So our main focus will be essentially on the molecules which are present in this serum and plasma in order to understand the immune system, because these are circulatory molecules which are present in the blood stream. And to identify these molecules, what are circulating, what are immune regulatory, we need to understand what are the content of plasma and serum to some extent. And therefore, we will oftenly discuss these two terms, plasma and serum, and therefore we need to understand this. So what is actual difference? I will emphasize here. The plasma, this portion is still the fluid part because we have added the anticoagulant such as EDTA, and here we don't have any anticoagulant. That means these are purely a clot form. So this is actually a clot of the blood, and what we are getting on the top of the clot is a small volume, what we call serum. So we can also call the liquid part excluding the clotting factor, containing again the antibodies and the immune proteins. That is what will be the content of serum. And coming to the two pillars of the immunity, which is again we will go in detail later on, but still to understand what are the things which are there in the blood, I already discussed. There are cellular part and there is a liquid part, right? And the liquid part we divide into plasma and serum. So again we have a cellular component which will be containing the cells of the immune system, and then we have a soluble mediator, so soluble components of the immune system such as protein, which are dissolved in plasma and serum. And these some examples are antibody, complement proteins, and the signaling molecules, which will essentially look into our further discussion. So understanding blood is very critical, because blood is the only fluid part of the human body that regulates the immune system, that because they carry a very specific all the cellular component and the soluble component in the form of liquid, and the cellular component in the form of cells also. So now we understood that the human body is constantly exposed to the pathogen, stress, and lot of environmental factors. Second, we understood that pathogens are in constant evolution process, so they are changing their proteins, they are doing mutations, and also the bacterial system where they can mutate the secretion system. Parallelly, we also understood that the host immune system is also evolving, where the pattern recognition receptors and the adaptive immune system in terms of memory is also evolving constantly in order to capture the immunity and see that they still fight with the mutation. And finally, we understood blood, which is a very critical fluid part of the immune system that carry all the essential component of the immune system. Let's understand the layer of different layers of the defense. So in this slide we can look into, there are specific 3 layers of the defense system, right? The first layer is called physical and chemical barriers, second is called innate immunity, and the third is called adaptive immunity. So these are 1, 2 and 3. So coming to the first seal, which is there on the very outside of the human body, and whenever there is a pathogen attack or any kind of a stressor attack, this comes at a very first layer that is targeted by that external pathogen. So the physical and chemical barrier which are there in the human body, what are the functions of these barriers? They prevent the microbial entry. That means they can breach, they can have a first layer which can stop the entry of anything coming to the human body. And what are those components? Skin. So skin is actually the largest organ of the human body, and this is present throughout the surface of the human body, and therefore anything entering into the human body comes through the skin. Unless until there is a cut or breach in the skin, we don't see any entry of the pathogen inside the human body. Second is epithelial surfaces such as mucus production in the alimentary canal. So when we are taking a food, we have always a mucus secretion. Mucus is a thick, viscous mediators which are present in the lining of the alimentary canal that can trap the pathogen or any kind of an entering substance that is not suitable for the human body. There are antimicrobial peptides. These are small amino acid stretch which are secreted by the human body in order to fight against the entering pathogens. And definitely the high pH of the stomach, the acidic pH of the stomach, which can also kill most of the unwanted moieties which are entering into the gut system. That means these are some of the things which are physically present in the human body, such as the skin, the epithelial surfaces, the hair follicles, the specific kind of mucus which are secreted in alimentary canal, the antimicrobial peptides, and the acidic pH. So these are some of the barriers which are there, which can curtail. If at all these are not sufficient to protect the body, then it comes the second layer, and the second layer is something which is present right from the birth, right? So we defined the second layer as innate immunity. Innate immunity is a term very specifically used for the immunity which is present right from the birth. That means a baby after birth getting this immunity from the mother. So mother can pass on the immunity, whatever immunity is there in the mother's body, that is transferred to the newborn baby. That means these babies which are newly born are not exposed to the environmental any environmental stresses or the pathogen. So still their body can fight with those pathogen if at all they are coming, because some of the molecules are carried through the mother's side into the baby's body. So this can rapidly detect and attack the pathogen which are entering into the human body, and therefore these are called innate. These are very, very conserved proteins which are helping to fight against the pathogen. And sometime, even though we have a first layer of a barrier, second layer of the innate immunity, we still have a problem, because the pathogens are smart enough to evade these two layers, and once they evade these two layers, they go to the third layer, and this third layer is actually the last stage of the immune system. And the final stage which is coming into the picture is called adaptive immunity, and this adaptive immunity is very specific. It recognizes the pathogen in a very synchronized fashion. We will understand in the next slide what do you mean, what are the difference between innate and adaptive immunity, but these are very specific. They can also generate long-term immunological memories. So which stands for the adaptive immune system can recognize a pathogen entering inside the human body as a foreign and will try to destroy it, and even after destroying, they can capture the memory of this destruction, so that in future, if at all this kind of pathogen is entering, the same kind of a memory will be immediately come into a process. And what we discussed, that we have the central dogma where the gene will start getting transcription to form messenger RNA, and this messenger RNA in turn will form a protein to fight against the pathogen. So this adaptive immunity is again a very, very critical part of the immune system. That means these three layers are constantly protecting us from the pathogen which are entering into the human body. And if at all there is a breach in all these three systems, the person will suffer from a disease condition. And we, as an infected person, the patient will start showing signs and symptoms of a particular disease condition, such as sneezing, cough, some kind of a diarrhea, or anything very specific to that, associated with that pathogen. That means this control mechanism is very important. Now, since we are discussing this innate and adaptive immunity, let's start understanding the comparison between the innate and adaptive immunity. What are the things which are important in order to understand and why these two are different? So as I said, that innate is very old. So evolutionary age, the first parameter is this is very ancient, innate immunity, while adaptive is gradually done in the human body post exposure to a environmental stressor such as pathogen, and therefore we can call it a recent adaptation of the pathogen exposure. As the innate immunity present right from the birth, and therefore once there is an entry of a pathogen, response is immediate. So within an hour of infection, we can see the immune response coming into picture, and we call it innate because it's already present in the human body. But the adaptive comes a little later because it will require certain response time, and therefore once the antigen or the pathogen exposed to the system, the adaptive immunity takes times to respond, and therefore sometimes it is days and sometimes it may be a week based on the pathogen type. So it's a slower response altogether, right? That raise after some days. Target recognition, as I discussed again, these two terms are coming into existence. Conserved structure. So the pathogen associated molecular patterns, which are very specific proteins or structure present in a pathogen, and the pattern recognition receptors are the very specific structure which recognizes the PAMPs present in the host cell. So PRR is host specific and PAMPs are specific to the pathogen. So these are conserved, but here the adaptive response is highly specific. So they can generate a diverse variety of antibodies, which we will see little later. So one which is entering inside the body is called antigen. The one which can generate the antibody, and therefore it is called antibody generator. Antigen stands for antibody generator, and these antigens are actually a foreign molecule. So these are very diverse in nature, and to fight with those antigens, every time we need a new antibody, and the host immune system can synchronize it in a very specific way. So these are very specific, very dynamic in order to fight against a changed antigen. We also looked into the memory. As I said, the innate immunity is right from the birth, so they don't understand the immunity, so they don't have any immune response. While in case of adaptive, we do have a memory response, and these can give a long-term protection against a pathogen. What are the components? Of course, I will not discuss a detail about these components, but let's try to understand few names here. The key components of the innate immune system is macrophages. Macrophages or macrophages are, macro stands for bigger and phages means eater. So these are the cells, the big cells which can eat a pathogen or eat an antigen. Neutrophils are the very specific granular cells present in red blood cell. So if you see, there are granulocyte and agranulocyte in the blood. One of the granulocyte is neutrophils. Then we have cells which can naturally kill anything. That's called natural killer cell, NK cells. And of course, there are soluble components present in the blood called complements. So these complement proteins and complements are also a part of innate immune system. That means these cells are actually present. They can fight immediately after entering a pathogen. They don't have an immune response, memory response, and of course they are very conserved in terms of their biological effect. While in case of adaptive, there are two very specific cells, B cell and T cell. We called it, the cells are present in the lymphocyte form. So lymphocyte is again a granulocyte. We call it granulocyte. Neutrophils are granulocyte, but lymphocytes are agranulocyte. They don't have granules inside the cytoplasm. So the B cell and the T cells are very specific lymphocyte which can raise against an entering pathogen. So these are part of adaptive immune system. The T cells are again divided into two types. One is called helper T cell and the cytotoxic T cell. We also call it TH and TC. So the cytotoxic T cell and the helper T cell population, which we'll see in the lectures, that how these specific T and B cells are recognizing pathogens and destroying it in a very synchronized fashion. So just summarizing the adaptive, that it's as I said, it's a recent, it's a very slow because it can generate a memory immune response slowly, and also these are very specific towards the type of antigen entering, and so it's giving a long-term memory response also. Okay, so coming to the next one, that we have now understood that the innate and adaptives are the two arms of the immune system, and we have pathogens which are entering into the body, and we look immunological cascade in a very different style. So suppose if we have a disease problem, so when there is a disease, that stands for we have a high pathogen burden. So when the pathogen amount is increased in the human body, we see the disease condition. And if the immune response, the immune control is very effective, then we won't see any response, any effect of the disease condition. That means this balance is very important, and to maintain this balance, we have lot of different mediators which are coming into picture. When I said the immunity is very useful, it is harmful sometime when we have an exaggerated immune response. So sometime it may happen that the immune response will be very high, and if there is a high immune response, then it could be deleterious to the human body, because it can start destroying its own cell, what we call autoimmunity. So autoimmunity stands for the term, auto means self and immunity stands for the immune response. That means the self immune response of the human body start destroying its own cell type because it is uncontrolled, and therefore there should be a balance between the immune response and the entering pathogen, and therefore these homeostasis are very important in the human body. these are some of the negative modifiers which we call it, and these modifiers are stress. Well, we always encounter with a lot of stress. Environmental pollutant are stress, the mental stress, the physical stress, the amount of the type of nutrient which we are taking, that will also give us stress. Of course, there are pathogens associated with this, and there could be some time when we see the immunosuppression. Immunosuppression stands for the lack of immunity, and this lack of immunity could be because of disease condition, because of nutrient status, or because of certain genetic disorders. And we could also discuss this later, that there are conditions in human body where the immune response is not generated in a proper way. The T and B cell, which we discussed in the adaptive cells are not generated in a proper way, and therefore when there is no immunity in a proper sense, we call it immunosuppression. So these are some of the negative modulator. What are the positive modifiers? And the positive modifiers of the immune system is vaccines. So we kind of educate the immune system to give pathogen weakened form of the pathogen. So suppose we are facing with a problem, and if somebody is not exposed to a pathogen, we are trying to give a weakened form of the pathogen so that the body immune system won't get the disease condition, and at the same time the body immunity can recognize this pathogen as a foreign and raise an immune response. So the body will be protected against the pathogen effect and also raise an immune response, what we call vaccines. And this vaccination is a very positive modifier which can build up the immune system. And of course, if there are the breach, we can also have some of the antibacterial or antiviral treatment, which can, or we have also antifungal treatment, antiparasitic. So the antiviral treatment could be a specific drug which can target to the viral say replication or the viral structure. Antibacterial, very well known antibacterial is, or antibiotic which we call, is penicillin. There are modifications which are existing in the market right now. So different forms of penicillins are there, and these all forms of the penicillin are actually used to curtail the infection. We have antifungal such as nystatin, amphotericin. These are very specifically targeting the fungus. And then we have antiparasitic as well, such as albendazole. These are target specific destruction drugs available in the market fighting against the different forms of pathogen. So finally, we would like to conclude understanding the defense dynamics, and this is very critical, that we understood that the host immune system is complex, and we need to understand the dynamics, how these are controlled. So the first point is evolutionary imperative, which stands for this immunity is shaped by millions of years of a strict microbial selection pressure. These are actually taking some of the very well conserved structure in the pathogen. So these are constantly evolving. There is a coordinated infrastructure which stands for a seamless integration of a specialized cell and lymphoid organs, which we'll see in the classes, further classes, that there are specialized cells and the lymphoid organs which are integrating in such a way that they can counterattack the entering pathogen. So when we read immunology, we can read immunology in two aspects. Either we can focus our attention towards a pathogen, and once we divert our attention towards the pathogen, we only fix our goal to highlight the changes in the pathogen, why these are causing disease, what are the changes that happen which is not evolutionarily conserved in the pathogen, what we could do in order to manifest those changes in a positive sense for the human population or for the human benefits. So, and then there is an entirely different area where we can focus entirely on the immune system. Then we understand that how we can modulate the immune system in a positive way. What are the things which we can do in order to raise an immune response? What are the things which can fight against the pathogen or entering microbes? And therefore, we see the two arms of the immunology. one, we focus on the pathogen side and we also focus on the immune side. But mostly in the immunology, we try to focus on the host side in order to understand the immune response. So as we also discussed that there is a dual capability. So there is a first line which is innate, and then there is an adaptive. So these are all the dynamic aspects which we need to understand. So these two arms are also there, and there is a strict regulation bound by an absolute requirement to maintain the immune balance and to prevent the host disease, and any deviation in this balance can be deleterious. So the immunity, high immunity can be leading to autoimmune disease condition, and lower immunity can lead to disease condition. So we should have a balance, a very specific balance of the immune system, and the deviation could be studied in a very different aspect when it comes to disease progression. So understanding these functions of functional logic principles in the foundation for advanced study in vaccine therapeutics and immunological diseases are very important. So gradually when we learn the problem in the immune system, we will see that how this deviation in the immune system could lead to deleterious effect when we see the disease conditions such as hyperimmunity or autoimmunity or any kind of other manifestation in deviation in the immunological responses. So these are all the dynamics of the defense system which we have in our body.